Good afternoon, everyone. I am Gloria Matthews of Oncosa Regenerative Therapeutics, and I'm actually here to talk to you about something that you might be surprised to see at a MedTech conference. This is a BLA pathway local liposomal Wnt protein for endogenous stem cell modulation. And the reason for that is that not only is it a rigorously studied therapeutic, but it has the potential to address a huge market in the MedTech space. This company was co-founded in 2015 by Sandy Madigan of Avalon BioVentures, who has decades of transactional experience, and by Jill Helms at Stanford, who has spent over 20 years researching this protein and this pathway. I joined in 2019, and I bring a strong clinical background, regulatory background, industry background, as well as deep R&D networks. We got an $18 million Series A in 2015. We've gotten $6.1 million in CIRM award funding and $12 million Series A extension in 2020 for clinical development when we opened our IND. We have just finished enrolling at the end of November of last year, our phase 1/2b study, and we're ready for pivotal stage. So why Wnt proteins? So these are, anytime you have an injury or disease in tissues, you need stem cells. And stem cells have to be told what to do in order to get the right type of tissue, whether it's nerve tissue, lung tissue, eye tissue, or bone. Because they're so important in the body, they have to be regulated very carefully. So Wnt proteins have to be protected from aqueous environments in order to remain stable in the body. Now, this makes them very hard to produce and purify. And Oncosa has developed proprietary methods to GMP manufacture and stabilize Wnt3A for clinical use by inserting the hydrophobic piece into the hydrophobic portion of liposomes and leaving the ligand portion out exposed in the environment so that it can target its receptor. So ART352L then is Wnt3A protein inserted into liposomal membranes. We have broad coverage of 5 patent families for this therapeutic, over 40 US and international patents issued with multiple pending. We have an aggressive IP strategy, and the portfolio currently has no obligations or restrictions. So you can imagine then that this, because it's a stem cell modulator, could have pretty significant platform potential. We, however, are not focusing on a platform at this time. We are focusing strictly on bone and more specifically on spinal fusion, hence MedTech conference. We know from our studies, as well as many studies across the globe over the last decade, that Wnt3A is a powerful bone former. We know that it activates bone-forming osteoblastic pathways. We know it inhibits bone-resorbing osteoclastic pathways. And we know that it drives osteogenic gene expression. But importantly, local delivery of this Wnt protein can be accomplished by taking bone autograft, soaking it in the protein solution, it just looks like an aqueous solution, and then putting it back into the body as you would for most orthopedic surgeries. This allows you to keep bone formation exactly where it's meant to be. I know this is busy, so bear with me. Here's why, because naturally in the body there are receptors for Wnt3A. They have funny names like frizzled and LRP56. And if you take with the rabbit picture, if you take bone graft and you soak it in the protein, or you have a control that has no bone graft in it, and then you look at what happens to the activity in the solution, you find that in the blue line on the top of the far right upper graph, that nothing happens, that the activity stays at 100% over 90 minutes. Whereas if you have bone graft, the yellow line, you see that it disappears from solution. And the reason it's doing that is it's being taken up by the bone graft cells. And we know that because in the lower right panel is a sample of some of the panel of osteogenic genes that we know this protein can then induce. It's very simple to put in the OR. It does not add surgery time. It does not add complexity. So harvest of bone autograft is standard for most orthopedic procedures involving bone. So bone graft is harvested. It's milled in the Stryker bone mill, which both we and Stryker have data keep the cells of the bone graft viable, which is important for gene expression. It's measured for a dosing basis, and then it's soaked for 30 minutes while the hardware is being implanted and other things are being done in the surgery. And the time it's needed back, it can stay there longer, and when it's needed back, it's replaced back in the body per standard of care by the surgeon. This is a huge market. We have focused on spine fusion based on a market analysis that we did back in 2020, showing that the lowest hanging fruit for us was spinal fusion. Our market opportunity at that time was about $3.7 billion. The interesting thing about this is even though it's the tough BLA path, the BLA path toughness comes from tackling GMP manufacture, getting your GLP tox in, making sure you get into IND, taking care of your assay release specifications and assay qualification, and getting into first in human patients. All of that has been done. So this is considerably de-risked. And the value, one value of having a BLA pathway, even though it takes a bit longer, is that if you get one with, for example, spinal fusion, there's so much rigorous data backing this up, both preclinically and clinically, that getting these other pieces bolted on is a lot easier than it is trying to get each of them de novo. The other facet to that is that the uptake clinically is much better, and also by reimbursement. Oh, one thing I want to point out on that, was talking about reimbursement, is if you look down there, these market opportunities consider the price of the product to be settled between $1,000 and $5,000. This is similar in concept to Medtronic's Infuse recombinant human BMP2, which is put on a collagen sponge and put into the spinal fusion area. This is the price point that they are currently getting, and they have reimbursement for this. Interesting, the cost of goods for this to manufacture is about $100. So the margins here are huge. We know from our studies in our labs, both at Stanford and at Ancasa, that this product is very efficacious in bone formation in all these models that you see here. Just put some schematics or some pictures just to give an idea. On the left in each of these pictures is the ART352L treated, and in the right is a representative of the PBS, the saline control treated. And I think you can see that there's a lot of bone, no bone, good, bad, purple, blue. I think it's clear that there are quite good efficacy signals. Those are all published as well, and I'm happy to provide publications to anyone who would like them. We also know from our models, both in aged rats and young rabbits, that we have efficacy in these fusion models. So as early as day 49 in the upper panels, you can see that the bone graft plus the Wnt drives much higher new bone versus initial bone formation. And in the young rabbits, who probably don't need Wnt3a to heal anyway, we see a dose-dependent increase in the number of sites, or the percentage of sites fused at 12 weeks. We embarked on a phase 1b/2a evaluation of this product in posterolateral spinal fusion. So we did single-level posterolateral fusion with a TLIF, a transforaminal lumbar interbody fusion, for spondylolisthesis, spondylosis, or stenosis. Only the posterolateral fusion site is treated with ART352L, and we assessed it 2 and 8 weeks, 6, 12, and 24 months for AEs, CT, fusion by CT, radiography for safety mostly, patient-reported outcomes for pain and fusion. And we had a robust data set with central radiology reads with 2 primary readers and 1 adjudicator. And as I said, that enrollment was complete in November 2023. And as COVID would have it, we have kind of a subset that initially enrolled, and then we have some later. So we actually have 1 and almost 2-year data on that early subset, and we're just getting the 1-year data on that last subset. We've had quite a bit of interaction with the FDA. I don't need to go through all of this, but we have comparability protocols established that they have accepted amongst all of the lots that we've used for tox, for this study, and for the next study. We also have agreement on the design of the sheep GLP toxin dose range finding study needed for the next study, and we have agreement on the phase 2b pivotal study. Our GMP manufacturing has been scalable up to 200 liter scale. I need to go through all these. We have over 600 vials from the current clinical study, over 3,000 vials from the new process, which is more potent and higher concentration. And we have, as I said, comparability. We have stability out to 5 years. This is the part that people cringe at when they're in the medtech world. This does take time, and we expect to be at BLA by 2029. The good thing is that most of the hard work has been done, and once we get through that, a smaller investment now works out to be a much bigger investment later, so I think it's worth the wait. Finally, we're asking for a $35 million Series B. We have the potential, we have a CIRM grant in right now for $15 million. If we get a $10 million co-fund, they will be very enthusiastic about that grant. That will allow us to complete the phase 2B and initiate 3, do the BLA preparation and GMP manufacturing that we need to do to get the phase 3 drug product. We could potentially initiate studies in other areas if that was of interest, and do clinical in-use optimization research. Thank you for your time.